So chapter 9, it's about biotechnology and DNA technology. So in this chapter, we're going to see what is biotechnology, what is genetic modification and recombinant DNA technology, and then we're going to see what are the roles of a clone, a vector in making recombinant DNA. So biotechnology, as a definition, is the use of microorganisms, cells, or cell components to make a product. And these products can be foods, antibiotics, vitamins, and enzymes, or modify these foods. Recombinant DNA technology, it's a branch of biotechnology. It is also known as rDNA. It is actually inserting or modifying genes to produce desired proteins. So that's the end product of this recombinant DNA technology, producing specific proteins. In order to do that, we have vectors. So a vector is a self-replicating DNA molecule that is used to transport foreign DNA into a cell. And this foreign DNA, it can be a plasmid or it can be a viral genome. Okay, so not necessarily has to be a plasmid. But no matter if it's coming, that DNA from a plasmid or a viral genome, it has to be self-replicating. That's the condition. So then when a cell takes up this vector DNA, the DNA multiply inside the cell, it can make a messenger RNA, and then you can have a protein coming from this messenger RNA as it is translated. So the cell then, when it's combined with this recombinant vector, can be grown in culture and forms a clone. And this clone will be identified because it has a specific phenotype that you can identify either with a color, in the case when you put a substrate that can be taken up at the cells, and then enzymes and that can transform this into a color. So the cell containing this recombinant DNA then can be used to harvest many copies of the gene of interest, and then you can make a library of your genes. An example will be human growth hormone, or ACG, AGC, that was inserted into E. coli. Another example would be the insulin gene that was inserted into E. coli. So with these genes inserted into E. coli, as these vectors were multiplying inside E. coli, you do recombination, and then you produce this AGC or insulin. So that's recombinant DNA. So an advantage of this is that you can do a lot of amounts of these proteins, and then they can be purified, of course, to be used. So you cannot plainly use these cells without purifying this product, or use this product without purifying it, and then that's cost effective. So here we have this illustration for this process. You have a bacteria that has the nucleoid DNA here, and it has this plasmid. This plasmid can be extracted from this bacteria, and then you have your gene of interest coming from your whole, the species that you want to study. In this case will be human, for instance, human DNA. And then you cut the piece from this human DNA that codes for human growth hormone. Then you cut the DNA, the vector DNA. You insert this gene into this DNA from the vector, and then you form a hybrid, a plasmid recombinant DNA.
And then in another cell that it didn't have any other plasmid, you inject this plasmid DNA that is recombinant. So this will be your transformed bacteria. And then you grow it in the lab. You purify it. After you grow different copies of this, you extract only this hormone, and then you use it in a human. And then we have different examples. So you can even make vaccines out of these. You can make enzymes that can soften the fabric, and then again you can use it as therapeutic drugs like insulin, human growth factor, human growth hormone. Okay, so what is the difference between biotechnology and RNA DNA technology?
Biotechnology is the use of microorganisms or like cells and their components to make an end product. And then our DNA is the modification of genes to make specific like proteins or products.
Mm-hmm. So this is a branch of this, and it is used for make proteins. In one sentence, describe how a vector and a clone are used.
A vector is the piece of DNA that you transfer the selected gene that you put. The vector is where you put the selected gene into, and the clones are, and the vector is used to, is put into the bacteria, and it is cloned, and bacteria replicates and produces clones of itself, and the clones are used to produce the needed protein.
Now let's move into knowing how we can select for these mutations or these vectors. And then we are also going to see the process of making them, like how we use restriction enzymes. And we're going to see the properties of vectors and describe the use of plasmid and viral vectors. You cannot use them for everything. They have specific uses. And we're going to see the steps of PCR. So, something that it is expected as you are transforming cells is that not all the cells will be able to pick up your gene of interest. And since you want to make sure that you're actually having what you need to, you have to select these clones that you produced. So one way to select is by adding a molecule in the media to be able for the bacteria to use it or not, depending on what genes you have in your vector. And then from there, you will find your vector of interest. It's like selective and differential media. Now, in mutations, you have mutagens that cause the mutations that might result for the microbe to have a desirable trait. So sometimes, and not only in microbes, you can use it in other microorganisms. You can induce these mutations, and then you can select for something that provides an advantage. So sometimes we have a specific areas where we want to produce a mutation. Maybe you want to activate a gene that is advantageous, like like a tumor suppressor gene. You might want to activate that gene if that gene wasn't active before, or if it was losing its expression and you want to express it more. So that's where we're going with medicine actually, guys. We're trying to do like what is called site-directed mutagenesis. And then that is targeted and giving a specific change in a gene. So you have available many enzymes that are co-restriction enzymes that can help you to cut DNA at a specific sites. And then in the case of these restriction enzymes, we have two types. And these enzymes, they can be useful according to the sequences of DNA that you have in your vector and also in your host DNA. So you have to know the actual sequence of your vector, and you need to know also the sequence from your host. So some of these enzymes were used by bacteria to destroy the bacteriophage DNA. And then we find those enzymes, and then we use them now to clone genes. Some of these can create blunt ends when they cut directly in between the nucleotides, and some of them, they can create a stagger end and a sticky end. Most of the times, you want this to happen. You want to have sticky ends because these sticky ends make your gene of interest to have more surface area for binding and to prevent this from falling off if your ligase enzyme, which is one of the enzymes that helps to bring together your gene and your vector, to put them together to bind them to not lose your gene. So sometimes your ligase might not be working pretty well, and then in this case, sticky ends, they still help without having a very efficient ligase. No matter if you have a blunt end or if you have a sticky end, you will have the DNA to be inserted into your DNA vector, and then you join this by a ligase. I guess I can play this. Let me play this animation. So it's like pieces of a puzzle. That was all. Okay, so here we have some examples of sequences identified and cut by these enzymes. And these restriction enzymes are named based on the species that they were discovered from. So BAM-H1, it comes from Bacillus amyloleucophacins. ECOR1 from E. coli, Hay3 from Haemophilus aegyptus, Hindi3 from Haemophilus influenza. And typically the Roman numeral means that it was either the first discovered, second, third, etc. So here we have the recognition site for BAMH1. So it cuts this site between two Gs. After this G or before this G is preceded by or followed by ATCC. And same in here, but this one it will be CCTA. So this is a palindrome. So you read in kind of opposite directions. And then here for E. coli, we have the cut between this G and A, and then this A has to be followed by A, T, T, and C, and same one here. So it will be A, T, T, C. So these two, and this other one as well, the Hindi3, they make sticky ends. So they create a staggered cut, like this. And then Haemophilus influenza 3, or Hay3, it makes blunt ends. It cuts between two Gs and two Cs. And again, you will have a selection of restriction enzymes based on the sequences in your host DNA and your vector. And there are programs that once you put this information into it, it will tell you, well, I mean, you will get, you can use all of these enzymes because you will have, in this case, you will have, like, for instance, for Hay3, you have Gs and Cs, two Gs and two Cs in this region. But what about, it will never happen, of course. It will happen because you will have more than likely GGs and CCs in one sequence. But let's say that you have a mutation and one organism never have these sequences. In this case, your enzyme won't cut. And then what if your gene of interest, like it has in the middle GG and CC? You don't want to cut it in half, because in this case you will not be able to produce your messenger RNA for that protein that you want to produce, and then you will have a dysfunctional protein. So and these programs not only tells you where you will have this action of this enzyme, so it will show you a map of your genome and where it will cut, but also it will give you like a table where it tells you, well, if you use this enzyme, you will end up with fragments of these many nucleotides, etc. And I'm pretty sure, I mean, that was when I started to do this genetic combination, but I'm pretty sure that nowadays these software programs are very efficient and gives you more information than sometimes what you need. So here we have an example then of how some enzymes can work. So let's see, which one is this? This is BAMH1. No? No, it's EcoR1. So in this case, we have this double-stranded DNA, and then we have GAATTC in here, and the palindrome in this other one, of course, that's how you expect it. And then if you use EcoR1, you will cut between G's and A's, and then you end up with this piece with sticky ends. And then if you have DNA from another source, you can insert it and then increase actually, like, or even if you use the same, or if you use a DNA from another source, you can religate and increase the size of your vector, and then you can have two EcoR1s sites, and then you can introduce a bigger gene or a gene that has the complementary sequence, and then you can ligate it, and then you can have your recombinant DNA, your RNA. So sometimes that's also something that I remember that it has been done. You can introduce EcoR1s into a vector that didn't have it before. All you need to do is having the right sequence. And when it comes to these bacterial plasmids, there is also like a limit in the number of base pairs that you can have, because of course you can try to introduce a big gene and you make this plasmid very big, but something that is for sure is that when you introduce these vectors that are modified with your gene, your bacteria, when you introduce it, may not replicate, and it may be lyse. So it has its limit to how big this plasmid DNA would be. So vectors are then molecules, or well, DNA that carry new DNA into the cellular cells. And again, these are properties. They have to be able to self-replicate. And plasmid and viruses can be used as vectors. One advantage of viral vectors versus plasmid DNA that we know that comes from bacterial vectors, is that actually viral vectors, they can carry larger genes. So if your gene of interest that you might be studying is large, you might want to use a viral vector. And then one thing about the circular vectors is that it protects the vector from destruction from the recipient. The recipient not only has these type 2 enzymes, but also has some enzymes like the exonucleases, so that the previous enzymes that we talk about are endonucleases. They cut the DNA in the middle, but the exonucleases cut in the end, the DNA. So these enzymes, the exonucleases, can destroy a vector that will be linear, so we don't have it. So we have the circular DNA. And then we have something that's called the shuttle vectors that exist in different species that can move the clone sequences among various organisms. So here we have PUC19. This is the name of one vector. As any other vector or plasmid DNA, it has an origin of replication that we call ORI. And then you start having upstream genes, that these ones can be genes that codes for maybe amino acid utilization, etc. But in here at least we have the AMP gene. This is for ampicillin resistance. And then in this area, we have a cassette of genes in which you have LAXI gene that codes for galactosidase enzyme that can break down galactose. And then we have 3 restriction enzyme sites like for HindI3, BAMH1, and EcoR1. So then you have options when it comes to using certain restriction enzymes. That's one of these vectors that is highly used, the PUB19. Let's go to the next part. Okay, now let's talk about PCR. So PCR, polymerase chain reaction, is the process of increasing or making multiple copies of DNA for analysis. So this PCR can make millions of copies within a few hours, and it is used to diagnose diseases or detecting pathogens. And then we have variations of this PCR. We have reverse transcription PCR, or we have real-time PCR as well. And in the case of reverse transcription PCR, instead of using DNA, uses messenger RNA as a template. So the machine that amplifies your copies of DNA, it's a thermocycler. You program this thermocycler. You have a period of priming, a period of extension, and a period of denaturation with different temperatures. Priming is usually at 60 degrees Celsius, extension at 72 degrees Celsius, and denaturation at 94 degrees Celsius. As the thermocycler is running for the period of time that you use it for, then you amplify your DNA. Let's see, let me go back. Okay, so part of the cocktail in your PCR reagents, you have to include DNA primers. And these DNA primers, they have to be complementary to the piece of DNA that you want to amplify, your target DNA. You have to have your enzyme, DNA polymerase, and you have to have the nucleotides. You have to have the ATP, the DCTP, DTTP, DGTP. And you have to have the right buffer to have this cocktail added into this thermocycler. And then the first step will be to actually separate the two strands of DNA. So you have to increase your temperature up to 94 degrees Celsius. Sometimes it can be a little bit more depending on your target sequence. Like if you have, for instance, a bunch of G's and C's in that target sequence, you have to increase your denaturation or melting point. And then once your two strands of DNA are separated, you drop the temperature to 60 degrees Celsius because you need to ligate your primer. So in this case, your primer has to come next to this, and then you ligate it with that lower temperature. And then after you prime your DNA, you stain your DNA, and then you undergo this same cycle over and over again until you reach the number of temperatures that you want. Who has done here work with this DNA polymer and this? Most of you, you're okay. That's good.
It's my favorite. I love DNA reactions.
Yeah, it's very nice to work with this. So then, again, you continue several cycles, and then you increase your number of copies. Okay, now let's move into techniques of genetic modification. So we're going to see 5 ways of getting DNA into a cell. We're going to describe how genomic libraries are made, and we're going to differentiate cDNA from synthetic DNA, And we're going to explain how to locate a clone, how to probe DNA and gene products. And we're going to see an example of using certain cells like E. coli, Saccharomyces cerevisiae, mammalian cells, and plant cells when it comes to cloning. We're going to see the advantages. So here are the five different ways to insert, the different ways to insert DNA into a cell. So the cells can be transformed. So you can use chemical treatments to transform the cells, so you can make the cells porous with chemicals, and then that porosity helps for the cells to take up external DNA. So you will make something that is called competent cells. Some of these cells that are competent cells can be commercially available, in which case you don't have to make them. But if you don't have them available commercially, you can make them by creating these pores in the plasma membrane, and then the cells can take up this DNA, and then you can actually, if you created your competent cells, you can freeze them and you can use them for later. Another way to make these cells competent is not by using chemicals, but also by creating pores in the membranes with electrical current. This is called electroporation. And you can also remove the cell walls from bacteria, and then that makes them to be more easily handled because you have only the plasma membrane, and that will create a proplast. A proplast will be kind of a circular cell, and then you will be able to fuse this cell with other cells. So in this case, we have proplast fusion. This is a couple of bacterial cells, one with this DNA, and this DNA was not part of this DNA from this other cell, of course. So these bacterial cells can be different, and then you remove from both of them the cell wall, you create proplast, you put them together in a solution that is treated with this polyethylene glycol, and then the plasma membranes fuse, and then you have recombinant DNA. And then you can put this cell into a bacterial cell that has cell wall, and then you will have your transformation. This is proplast fusion. In the case of inserting foreign DNA to the cells, you have the DNA inserted into a cell by a gene gun, and then you can microinject these cells with a micropipette, a tiny micropipette. So here we have this gene gun that has DNA coated cells, and then you can inject them. And then here we have a microinjection of DNA into a cell. So this is the micropipette. You have here a device that creates a little bit of suction to keep this cell from moving, and this is the needle, and then you can inject your DNA. This is one of the techniques that is not very easy to do. You have to have a lot of steady hand for doing that, and you have to control the vacuum to prevent damaging these cells. And no matter if you use the gene gun or this, or either the proplast fusion, you always have to make sure that you have injected your DNA into the recipient cell. So you have to do some sort of growth of these new cells in media, and also phenotypic analysis, and then extraction of this DNA as well. So when you have then these insertions of these genes into a recipient cell, you can extract these cells from these cells, these genes. You can either use the same restriction enzymes to take away this DNA from your from your plasmids or your vectors, and then you can end up with several DNA fragments, and you can create your library. And maybe you want to keep a piece of the vector or not, depending on how you want to make your library with only purified genes or not. But you can have a clone for every existing gene in the organism if you want to, and then you will have your gene library. So this is a genomic library. So you will have this genome, in this case from this phage DNA, and you can have it from as well from this recombinant DNA plasmid. So what you will have within these regions of this DNA, these areas that you can cut with a restriction enzyme. And then once you cut it, you can form either a plasmid library or a phage library. So you can select for specific genes, like in this case, if they were color-coded, like the red gene library, the orange, the green phage library. So you just cut it, put it into a new plasmid or a new phage, and then you put them in your test tubes, freeze it, and then keep it for a long time. So when it comes to cDNA, complementary DNA is made from messenger RNA by reverse transcriptase. This one can be used for obtaining eukaryotic genes, and in this case, because the eukaryotic DNA has introns, do not code for protein. Messenger RNA will have the introns that needs to be removed, and then this will code only for the protein product. So, if you want to make your cDNA library from your eukaryotic DNA. So in this eukaryotic DNA, we have, of course, exon and introns. As you make your RNA transcript or your RNA polymerase, this will still have these introns and exons. When you splice these, you remove the introns and you only have your exons. So this is your messenger RNA. And then once the messenger RNA exits the nucleus through the nuclear pores, you can synthesize your first strand of DNA, and then your messenger RNA can be digested by this reverse transcriptase enzyme, and then you have your cDNA without the introns. So that's the difference between these eukaryotic genes. So you will have a copy of DNA without these introns when you use your reverse transcriptase. Now you can also make synthetic DNA, which is basically making a gene in vitro using a DNA synthesis machine, in which you have the sequence of the gene that you will have to know before, of course, and you will have all your oligonucleotides, just like if you were doing a PCR, that will be synthesized and joined according to how you will be adding them into this machine. So this is a DNA synthesis machine. So, contrast the 5 ways of putting DNA into a cell. What are the 5 ways?
Transformation,
Mm-hmm.
electroporation,
Mm-hmm.
prototransfusion,
Mm-hmm.
microinjection,
Gene
Okay. So, what is the purpose of a genomic library? Why would you want to have genes from an organism? Would you care to know the function of those genes? Like, what does this gene do? Does it prevent from having cancer or help to utilize certain amino acids, code for an enzyme? And then, why isn't cDNA synthetic?
because it is synthesized based on the original cell's mRNA, and it's just using reverse transcriptase enzyme to make it.
Very good. Okay, so